Howdy, and welcome to the Unit on Protein Function Part 2, the structure and function of hemoglobin and also myoglobin. These two oxygen binding proteins have similarities and differences in how they bind and function with oxygen. And I want you to introduce yourself to hemoglobin by visiting Hank Green's Crash Course video, Respiration Part 2, because this is going to be a good overview for understanding how hemoglobin works, how its structure is linked to its function. Its function is to bind oxygen and transport it to the tissue, and then let go of the oxygen so that your cells can function. Okay, so your cells respire, they live on oxygen, and they need to get that oxygen from your lungs, and that's the job of hemoglobin. Learn about that from Hank Green. All right, so in this first part of part 2, we're going to talk about hemoglobin and myoglobin as an overview, and the bullet points listed here are going to get broken down as we move on through part 2. Myoglobin is a single polypeptide, so it has tertiary structure but not quaternary structure, and it has a single binding site for a heme prosthetic group. That's a fancy way of saying it binds a non-protein component called heme, and heme stays bound to myoglobin like a prosthetic leg would be bound if you were walking, okay, and you needed a prosthesis. The heme binds to the oxygen, and when the oxygen concentration is increased, you see more binding to more myoglobin in solution, and that gives you what we call a hyperbolic oxygen binding curve. So watch for that. Hemoglobin, by contrast, even though it also binds oxygen, and it binds to 4 heme prosthetic groups, one for each of the 4 polypeptides, so this guy has quaternary structure, it can adopt 2 different conformations. Without any oxygen, it's called deoxy, or the T tense state, and when bound to oxygen, it is called the oxy, or relaxed state. R stands for relaxed. And the 2 different conformations have different binding affinities. The T state has a very hard time binding to oxygen, and the R state binds oxygen very easily. So we're going to see how that affects the shape of the binding curve. Here's a hint. It's very different from the myoglobin binding curve. What's going to happen, as you'll see, is that oxygen binding triggers a conformational change in the hemoglobin. We just talked about a conformational change in the flu virus hemagglutinin that allows it to function, and that's not an uncommon thing. Remember, proteins have to move to function, and this conformational change is very small, but it's big enough so that when hemoglobin binds to oxygen, each one of these subunits interacts with the other subunits, making the switch from the T state to the R state very rapid. And so you go from it's either an all or nothing binding to oxygen. So you go from no oxygen bound, and then when it's triggered at a certain concentration of oxygen, then it goes to the R state, and all of the subunits of the tetramer bind oxygen. That's called cooperativity. Cooperativity is when the individual components of a quaternary structure of a protein all act together, and that gives you a sigmoidal, or S-shaped, and you'll remember S-shaped because sigmoidal begins with S, binding curve when it comes to oxygen binding. We're going to talk about regulation of oxygen binding to hemoglobin. There is no regulation of oxygen binding to myoglobin, so it will only be in the context of hemoglobin. And a big hint here, all of the regulation that's going on for oxygen binding to hemoglobin are in place to help release the oxygen back to the tissues. Okay, so we're going to watch how the Bohr effect and BPG lower hemoglobin's affinity for oxygen and how it does that. And then we'll talk about mutations that change hemoglobin's oxygen binding properties and even cause disease in one example. All right, so this is myoglobin. This is another Irving Geis drawing. He's my favorite scientific artist. And what you're looking at in Irving Geis's beautiful drawing is the nomenclature for labeling parts of the fold of myoglobin. All right, so the myoglobin structure has an A helix, an AB connector, a B helix, a BC connector, which is behind, a C helix, a CD connector, and so on. EF, F prime, FG, GH, and H, and the C terminus. You don't need to memorize the structure of myoglobin, but what you see here for myoglobin has implications for the nomenclature of important binding residues in myoglobin and in hemoglobin. Okay, so that's why I'm introducing that concept of, for example, the H helix, which you're going to see again, and the F helix, etc. Now, it turns out that, here's another Irving Geis, hemoglobin has the same structure in each of its individual subunits as myoglobin. So we've zoomed out a bit, and now we're looking at the tetramer. So the hemoglobin, remember, has 4 polypeptide chains, 2 beta hemoglobin chains and 2 alpha hemoglobin chains. And that means that it is a heterotetramer. And up at the top, you're looking at the heterotetramer in the deoxy state, and at the bottom, you're looking at it in the oxy state. And in both states, at first glance, each one of the monomers, so this would be one monomer of hemoglobin, resemble, and in fact, the fold is identical to the myoglobin structure. Okay, so all of that nomenclature with F helix and H helix, etc., is going to apply to hemoglobin as well. And like myoglobin, there is a heme prosthetic group that binds. So if I could go back one slide, the heme prosthetic group is this iron-containing group here, and that would be the iron right in the middle, bound to that organic honeycomb-shaped planar heme. Okay, so there's one heme binding site on the single polypeptide of myoglobin, and 4 heme binding sites on the 4 polypeptides of the tetramer of hemoglobin. Now, if we look carefully, and I've circled these sites to highlight them, and also the artist, Geis, has used an arrow in the axis of symmetry, which is that eye-shaped axis in the middle. There's a double-headed arrow there indicating that there's a distance change in the center of the deoxy and oxy conformations. So this is the T state, the deoxy state, and it's got a wider gap in the middle. And that becomes important when we start binding other molecules to hemoglobin. That gap narrows as the protein irises down. Maybe you can see those ghosted gray arrows on top of the oxyhemoglobin, indicating that the 2 sides of the hemoglobin tetramer are twisting against each other, just as if you turned a jar on its side and you tried to unscrew the cap. That's the type of conformational change that's going on with hemoglobin. When oxygen binds, it not only twists the 2 monomers on this side with respect to the 2 monomers on this side, but it also breaks an important non-covalent bond. So there's a non-covalent ionic interaction present within this circle, as well as a good packing interaction. And that good packing interaction and salt bridge, which is another term for ionic interaction, is broken when you twist the hemoglobin when you go from the deoxy to the oxy state. So when When oxygen binds, you get this rotation, opening the jar, taking the jar lid, unscrewing the jar lid, actually moves the positions of the sort of the elbow of deoxy, moves down, away from its position when it was upward in deoxy. So those are circled on both sides of the molecule because this is a symmetric molecule. It's also happening on the back side. Okay, so that breaks a salt bridge in the R state, and that very subtle conformational change changes the affinity of the molecule for oxygen. So this is the change that we're going to be talking about when we talk about oxygen binding to the iron, which is just that white dot right in the middle of that flat planar heme group. Okay, and again, there are 4 of those, even though you can only see 2 in this picture. All right, so the fact that hemoglobin undergoes a conformational change that affects its affinity for oxygen means that it can be regulated, and it has what's called allostery. We're going to talk about allosterism more when we start talking about the regulation of oxygen binding to hemoglobin. But allosteric proteins are proteins that have regulatory sites like on and off switches, outside of their normal binding site. Okay, so we'll talk about how allosteric effectors, molecules like BPG or a proton or even a carbon dioxide, can bind to the hemoglobin tetramer and change its function. So by binding to a site other than its active site or its oxygen binding site, in this case, you are attaching a molecule to another site, allo other. Okay, so that's why we study hemoglobin and myoglobin. It is the hallmark of allosterism. Okay, now I'm glad we got that out of the way because we're going to see that coming up. All right, so this slide is going to function on the difference in is going to focus on the difference in the functions of these proteins. So they both bind oxygen, but the job of hemoglobin is to transport oxygen. It's going to bind it in the lung artery interface and transport it and release it in the tissues, whereas myoglobin's job is to store oxygen in the muscles for fight or flight. Okay, so it's going to hold on tightly to oxygen until there's an emergency, whereas hemoglobin has to be able to release oxygen, so it has to bind more loosely and release it every second of the day. Okay, so we're going to look at the oxygen binding curves for these 2 proteins, keeping in mind they have these different functions. We already talked about how myoglobin, which is abbreviated capital M, lowercase b, is monomeric, single polypeptide chain. Hemoglobin is tetrameric. It has 4 polypeptide chains, 2 alpha chains, 2 beta chains. Myoglobin is about 153 amino acids, not a big protein, it has a small molecular weight. And the alpha and beta chains, while different in sequence from each other and from myoglobin, fold just like myoglobin. Okay. All right, so now I want you to really pay attention to this curve because you may have to explain it on an exam. All right, what we're looking at here is myoglobin binding to oxygen and hemoglobin binding to oxygen. On the x-axis, we have increasing concentrations of oxygen expressed as the partial pressure of oxygen, or PO2 in pressure, torr, units of torr. This partial pressure is explained in Hank Green's crash course video, so watch that if you want to understand the relationship between partial pressure and just straight off concentrations of oxygen. Low concentration, high concentration. On the y-axis, we're looking at percent saturation. That is, how many of the ions in the hemes of the subunits of hemoglobin or on the heme of myoglobin are bound by oxygen. So you're going to have in a test tube hundreds and hundreds upon thousands of hemoglobin molecules, and in another test tube hundreds of thousands of molecules of myoglobin molecules, and at a certain percentage of saturation, some of those molecules are bound to oxygen and some of them are not. That's what that means. All right, so no binding, 100% binding. All right, now we're ready to distinguish the curves. The myoglobin curve is the hyperbolic curve in blue, and the hemoglobin curve is the S-shaped curve called sigmoidal in purple. If we start at high oxygen concentrations, and that's why I have this arrow over here, such as at the lung artery interface, the concentration of oxygen at the lung artery interface is 100 torr. That's high. And you see that both myoglobin and hemoglobin are fully saturated. They're loaded up with oxygen. And then as you go lower and lower in oxygen concentration, pay attention to the blue curve, it still stays saturated with oxygen. That is, all of the molecules in your muscles stay bound to oxygen until you start really working your muscle. And as you work your muscles more intensely, for example, in the gym or if you're in a fight or flight situation, you're running out of oxygen, and so you're going to very low oxygen concentrations, and under those extreme conditions, the myoglobin will give up the oxygen and help you out. Okay, so that's why it does let go at very low oxygen concentrations. Hemoglobin's job is different. Hemoglobin picks up oxygens at the lung artery interface at about 100 torr, and then it starts to deliver your oxygen at the vein capillary interface, right about 30 torr. Okay, so this is still where you're resting. You're not, fighting or fleeing. This dotted line indicates really the oxygen concentration at the tissue vein interface, where those capillaries meet up with the tissue and start to feed your tissue oxygen. And so hemoglobin needs to let go of the oxygen, and that's what it's doing. It's starting to give off oxygen, and that's what gives rise to this this rapid transition over a narrow range of oxygen concentration. It gives up the oxygen. Okay, so oxygen transport, oxygen storage. Those are the different functions of those molecules, and that's why they have different binding curves. All right, so we're looking now at the structures and in a ribbon diagram, and each polypeptide has a different color ribbon. And so myoglobin is shown, it's all green, so that is your monomeric protein with a single heme group, and it is all alpha helices. So this is an all alpha protein, myoglobin. Hemoglobin is also an all alpha protein, but you see there are 4 different colors, so that's the tetrameric quaternary structure that you have in hemoglobin that you don't have in myoglobin. You'll see in textbooks that hemoglobin is described as a dimer of dimers. So yes, it's a heterotetramer, but each alpha beta dimer docks against another alpha beta dimer, so that's the dimer of dimers. So I just wanted to shout that out in case you read that in the textbook. Central to the oxygen binding ability of both proteins is the porphyrin ring that houses the metal ion iron. Iron in its +2 state is ferrous iron, and ferrous iron can coordinate oxygen. This is protoporphyrin 9. That's just called protoporphyrin 9 in the absence of the iron. As soon as you load it up with iron coordinated in the middle, coordinated by all those nice nitrogens on those protoporphyrin rings, these look like histidine, imidazole rings, right? It's part of the imidazole protoporphyrin family. those can coordinate iron and hold it in the center of the protoporphyrin ring, and now you have heme. So heme is the iron-loaded protoporphyrin ring. You will not be expected to draw the heme. Just remember ferrous iron, Fe2 plus, sometimes Fe parentheses, Roman numeral 2 and parentheses. Okay. It turns out that helix F, remember I told you the helices were labeled by letters back in the day. The first structure that was solved was myoglobin. We don't do that anymore, but its nomenclature stuck. So histidine in helix F is the 8th amino acid in that helix. So it's called histidine F8, and histidine F8 coordinates the iron on the protoporphyrin ring. Let's go back. 4 coordination sites are provided from the protoporphyrin 9 ring, but ferrous iron actually has 6 coordination positions. So the 5th coordinating nitrogen is coming from the imidazole ring of the histidine, position 8 on helix F. Okay? That's the 5th ligand. Ligand is a fancy word for binding entity. Okay, so the 5th ligand for the iron is histidine F8, and the oxygen molecule is the 6th ligand. Okay, so in deoxyhemoglobin, the ferrous iron is coordinated by 5 ligands, 4 of them from the porphyrin ring, and 1 of them from histidine F8, and then the iron in oxyhemoglobin coordinates to the oxygen molecule as its 6th ligand. The alignment of the O2 molecule is tilted relative to the perpendicular line to the heme plane, so that's what you're going to expect to see on the slide. And that's down here. So O2 is shown at the bottom. That's the 6th ligand, 6. And it's coordinating the heme iron, the ferrous iron, Fe, in the middle, from below. And then there's histidine F8 coordinating the iron from above, and then the other 4 ligands from protoporphyrin 9, part of the heme, are coordinating in that plane. Okay, so I want you to keep this geometry in mind, because the conformational change, that twisting of the jar lid, is going to affect the way that these that the histidine F8 interacts with the ferrous iron when the oxygen binds or not. All right, so when it comes to myoglobin also binds to the heme iron and coordinates it in exactly the same way. And so it's also ferrous iron, and then the 6th ligand is oxygen that binds at a tilt, just like in the last slide. If the iron is oxidized to form the 3 plus iron state, then it's ferric iron, and ferric iron can't bind oxygen. Myoglobin with ferric iron is called metmyoglobin and can't bind oxygen. Hemoglobin with ferric iron is methemoglobin, and it can't bind to oxygen. There are enzymes that can reconvert methemoglobin and metmyoglobin so that they will coordinate the ferrous iron. They'll let go of this one and coordinate the ferrous one. But those enzymes are absent in people with methemoglobinemia. And there's a picture of that, a family. I include it on the eCampus website as well. So this is the Fugit family, and the Fugit family in Kentucky was characterized by blue family members. Their skin was blue because their iron was in the ferric state in abundance and could not bind oxygen. And if you remember, our veins are blue because they're transporting actually T state hemoglobin, deoxyhemoglobin, back up to the heart, right? For the artery to pick up some more oxygen at the lung artery interface, where they will bind to ferrous. The ferrous oxygen will bind to the ferrous iron, of course, will bind to the oxygen, and that will make your blood red. Okay. Well, these people don't have as much red blood, so they have more blue blood, and it shows through their skin. And what happened in the family in this mountain part of Kentucky was a little bit of inbreeding. That's what you're seeing right here is Martin Fugett's wife's sister married one of their sons, and they had 4 blue children, and that's the picture that you're seeing here. Here are the 4 blue children. This is Zach Fugett, and there were some more blue children later on in their family, and there's even been I think another blue person came up more recently. But as there's been less inbreeding, this phenotype is recessive, so it hasn't shown up as much anymore. So it's an old phenomenon. Methemoglobinemia. All right, we'll start the next video here, where we'll talk about the changes in the conformation of the heme iron upon oxygen binding.